Hello friends. So, hello friends, I'm Ishu Gupta and I'm here to teach you biochemistry in a very short way. So, let's begin with the amino acids generally found in the form of 2-amino carboxylic acid. As we can see in this structure, this is the carbon 1, this is the carbon 1, this is the carbon 2. So, 2-amino carboxylic acid, amino acids, they generally exist in deuterine form at neutral pH. As we know, amino acids fulfills various functions in the organism, and they also serves as the component of peptides and proteins. Here is a minute difference between the peptide and the protein. Are the polypeptides contain more than 50 amino residues, and peptide are, or we can say 2, 3, or 3 to 10 consist these amino acid residue. For example, if it contains 2 amino acid residue, then it will call a dipeptide. Similarly, with tripeptide, it contains 3 amino acid residue joined by amide bond. Amide bond. So let's discuss with this amide bond. Supposedly, I had taken one amino acid here, and one is by removal of H2O, it will form a amide bond. This is the amide bond. Come to the point. There are 2 types of amino acids. Proteogenic and non-proteogenic. Proteogenic amino acids are included in the genetic code and therefore regularly found in the proteins, in which some of them undergo further change following their incorporation into the proteins. Non-proteinaceous amino acids are those one which doesn't include in genetic genetic coding, but they also derived from proteogenic amino acids by certain changes uh, like transformation. We already talked about that amino acids are the main component of peptide and proteins, and there is this amino acids are also the main component of lipid, phospholipid, That is serine, a neutral amino acid. And glycine in bile salts. So अभी इसके फंक्शंस डिस्कस करते हैं कि अमीनो एसिड्स के क्या क्या फंक्शंस होते हैं जो प्ले करता है द वाइटल रोल. सो some functions are neurotransmitter, some amino acids function as neurotransmitter themselves, and others are the precursor of neurotransmitters, mediators, and hormones. Like, for example, glucose in glucogenesis, heme, and for purine and pyrimidine bases of all other molecules. Amino acids are the main component of food as well. So let's
go through this diagram. Here is our group, that is a side chain of any amino acids that will vary. Here is the carboxylic acid group. Here is the amino group. Here is the hydrogen. So, as we discussed earlier, the amino acids are the main component of peptides, protein, and phospholipids in the form of serine, that is a neutral amino acid, and it is act as a precursor of keto acids, biogenic amines like serine, cysteine, threonine, aspartate. And what are the function of this serine is to activate glutamate, that will functions as a neurotransmitter. Glucose for glucogenesis nucleotide, that is purine and pyrimidine bases plus heme and creatine. It will also function as a neurotransmitter. In which glycine, glutamate and aspartate are used. Glutamate contains gamma aminoglutarate that will function as a neurotransmitter that will pass the signal from one neuron to another neuron cell. So let's talk about the optical activity of amino acids. So as we know that all the amino acids are mainly alpha amino acids, because this carbon is the alpha carbon and this group is attached to the alpha carbon. That is why it is called alpha amino acids. In contrast to beta amino acids such as beta alanine and taurin. These are found naturally in the environment along with alpha amino acids. Most alpha amino acids have 4 different substituents, like in this diagram, 4 different substituents Therefore, this carbon would be a chiral carbon. This carbon would be a chiral carbon and form two different enantiomers like D and L amino acids. Among the proteogenic amino acids, I told you about the proteogenic that will take part in genetic coding. So, except in all proteogenic amino acids, glycine is not chiral. Mostly in nature, L amino acids are found, but in bacteria, some D, but in bacteria some antibodies contain D amino acids. For example, in bacteria murin is present. In animal metabolism, D amino acids would disturb the enzymatic reactions of L amino acids. D disturb करते हैं. That is why they all broken down in liver by the enzyme D amino acid oxidase. The Fischer projection is used to present the formula of chiral centers in biomolecules. This one. This is the Fischer projection. It is derived from their three-dimensional structure as follows. For example, what happened? सबसे पहले क्या होता है इसमें? ये जो टेट्राहेड्रन है, this one, इस तरह से रोटेट करता है कि सारे जो ग्रुप हैं वो ऑक्सीडाइज हो जाते हैं टॉप पे. In such a way, rotation is then continued until the line connecting line this is the pair of enantiomers. In L amino acids, this group is on left side, while in D amino acids, this group is on right side. Mirror image of one another, but not superimposable. This is the meaning of enantiomers. So, thank you so much for watching this lecture.